Ladies and gentlemen, welcome to the 2013 Peter Baker Lecture. As many of you know, Peter Baker was one of my predecessors, eminent predecessor, as head of physiology here at King's, and died absurdly young in 1987 in Harness. And he was a great scientist, and we have an annual lecture in his memory. And I'm very glad that Phyllis Baker, his wife, is here as usual with us. And I'm delighted to introduce this year's speaker, Professor Fiona Watt, who is director of the stem cell and I've forgotten the remains of the title now.
Sorry, stem cells.
Well, the Stem Cell Centre at King's. And she started off in Oxford, where she did her DPhil, worked in MIT in the US, then the Kennedy Institute of Rheumatology in London, London Research Institute, and became Deputy Director of the Cancer Research UK Research Institute and Deputy Director of the Wellcome Trust, Sanger Centre for Stem Cell Research. It was a great coup for King's to attract Fiona here to run our stem cell unit, and there's little more to say other than to say thank you for accepting it.
Oh. No, don't tell me. I can do it.
Sorry.
Done it. Okay. Well, thanks very much. I'm really delighted to give this lecture, and you know, as we all age, to realise you know how many years we lost of your husband's life is really, it's sad. It's a shame. So I'm going to tell you about the work that we do in my lab at the moment, and I'm going to give you an overview of why I think stem cells are interesting, and then I'm deliberately picking very different aspects of our work to illustrate the sorts of things that we're interested in. So this simple schematic really gives you a generic introduction to why people are interested in stem cells. These can be defined as essentially any cell which has the ability to make more cells like itself, which is called self-renewal and is shown by this arrow, or to produce daughter cells that undergo a further maturation or differentiation. And it was realized many years ago that the body had populations of stem cells when people started analyzing the blood and realized that every day our body needs to produce very large numbers of red blood cells which don't have a nucleus, and so they can't replenish themselves. And of course, since then, the concept has been applied not just to adult tissues, but to embryonic cells. And there is a great interest now in the potential of different kinds of stem cells for use to repair damaged tissue. So stem cells make more stem cells, and they produce more mature or differentiated cells. And the question is, what controls those decisions? So in part, what a stem cell does depends on its intrinsic nature. So if a stem cell is in an embryo, it's going to do different things to if it's in the bone marrow and making blood cells, for example. It also, though, depends on signals that it receives from its external environment, which is usually referred to as the stem cell niche or microenvironment. And the kinds of signals that we know about here could be contact with neighboring cells. It might be physical factors, such as the stiffness of the tissue. So these extrinsic signals are quite varied. And in addition, we appreciate that this interaction with the niche is not simply the stem cells being instructed to do things by these external stimuli, because we know that stem cells, often in response to these signals, actually remodel the niche. So when we think about stem cells in the context of a tissue, we're always thinking about how different populations of cells communicate with one another. Now, many years ago, when I was a postdoc at MIT, I started working on the epidermis, which is the outer covering of the skin. And so in this talk, I'm going to be talking about intrinsic and extrinsic signals that control stem cell behavior. But I'm going to start off with a very stripped down approach which is based on studying the properties of individual human epidermal cells in culture. And then I'll go really in the other direction and tell you about some of our mouse models in which we are manipulating the epidermis or the underlying connective tissue and looking at how the tissue responds. So this gives you an overview of the different regions of the skin that we're interested in. And this diagram is based on the structure of mouse skin. So in the epidermis, we have a multi-layered epithelium called the interfollicular epidermis. And its function really is that the stem cells in the basal layer should produce differentiated progeny that form the dead layers of your skin, the outermost barrier of the skin. And as these cells are shed from the surface of the skin, they're replaced through proliferation of stem cells in the basal layer. We also, of course, particularly prominent in mice, we have the hair follicles and the sebaceous glands. They're all maintained by epidermal stem cells. And not shown on this slide, we know that the epidermis has stem cells that make the sweat glands. And then, in addition, the epidermis is separated from the underlying connective tissue, or dermis, by a specialized extracellular matrix called the basement membrane, which is shown here in blue. So when I went to MIT many years ago, I was attracted by the fact that it was possible to isolate cells from normal, healthy human skin and expand the cells in culture. And they would spontaneously make a multi-layered sheet. And within a very short space of time, less than 10 years, this technology for growing and expanding cells from human epidermis ended up being used in the clinic, where somebody had been badly injured, badly burnt. Their small biopsy of healthy cells were taken, expanded, and then grafted back onto the patient. And so we knew from this work that stem cells, which we're interested in, must persist in culture. And what I liked then and still like very much about the epidermis is that if you take away the hair follicles and the connective tissue, you have a beautifully simple tissue which is arranged as stacks of cells, and you could think of it as a bit like a brick wall. So in this diagram, we're just focusing on the interfollicular epidermis, and the markers that I'm showing you relate to human epidermis. So we've got the stem cells in here in the basal layer attached to the basement membrane. We know the cells make more stem cells, and then they produce daughter cells which stop dividing, and then they move out of the basal layer and undergo this process of terminal differentiation to make the skin barrier. And over the years, my lab and many other labs have put effort into identifying protein markers of different stages in this process of differentiation, some of which are listed down here. And we've put also some effort into finding cell surface markers which would help us define or enrich the stem cell population. So this is a beautiful, simple system. If you can grow human epidermis in culture, you know that you protect the stem cells. You know that as the cells differentiate, they build this multi-layered structure. Then you can start to really hone in on the interplay between those extrinsic signals that I told you about and intrinsic signals. So when I was in Cambridge, we decided that one approach we would take is to make what we describe as micro-patterned islands, which would allow us to control the external environment of single cells very precisely. And the strategy that we used, this is a collaboration with Willem Huck's group, was to take gold-coated glass slides, take a rubber stamp, and use this stamp to imprint a chemical onto the surface of the slide. And then we use this chemical initiator to grow polymer brushes of a compound called poegma. But what's important for this is that poegma brushes are protein resistant. They're used, for example, to prevent barnacles building up on the hulls of ships. But in our application, we then take these substrates and we immerse them in a solution of extracellular matrix like collagen. And the protein passively adsorbs to the exposed gold, but it doesn't adhere to the polymer brushes. So now if we plate cells and selectively capture the stem cells, we can control the way they spread and the shape they adopt. This shows you two examples. These are scanning electron micrographs of single cells captured on circular islands of 20 micron diameter or 50 micron diameters. And so you can see that when the cells are on these small islands, they're attached but they're not spread, and on the large islands they spread completely. What we did then was to plate cells on a range of these different islands, wait for 24 hours, and for each given island area we quantitated what proportion of the cells had begun to differentiate and were expressing markers like this protein involucrin. So this first experiment showed us that if a cell lands on an island like this, it's much more likely to decide to undergo differentiation than if it lands on an island like this, where it can spread completely. Because this setup is so simple, we were able to identify the signaling pathway by which the restricted cell spreading results in the decision of the cell to differentiate. And this work is published, so I'm just going to summarize what we found. So here's our single human epidermal stem cell here, and here are 3 conditions. If the cell spreads completely, it doesn't differentiate. If it fails to spread, but we don't provide external growth factors, it also fails to differentiate. In order for the cell to differentiate in this admittedly very artificial environment, we need the combination of the cell being rounded, and we need the presence of soluble growth factors. And what happens then is that we get activation of a program of gene transcription, which is mediated by this factor here, SRF, or serum response factor. This leads to increased transcription of further transcription factors called AP1 factors, which in turn regulate many of the genes which are expressed during differentiation. We know that the ability of the cell to activate the SRF pathway depends on the state of assembly of the actin cytoskeleton, because this controls whether this cofactor MAL is in the cytoplasm or in the nucleus. So it's very gratifying, really, to be able to take single cells and identify precisely how it is they're responding to external signals and executing a differentiation program. So from those core experiments, we've really expanded our work in a number of different ways. Gernot Walko, who's here in the audience, is doing some very nice work in collaboration with Michael Linden and Elske Henkerts, making live cell reporters of those decisions. But I want to show you next other approaches that we're taking, which is to go back and have a look at the histology of human epidermis and say, well, you know what? It's not single cells sitting on glass. Actually, the cells are together. The stiffness of the underlying connective tissue is much softer than glass, and the interface between the epidermis and the dermis undulates. So Wesley Chua, who's in the lab, is modeling this undulation process using different sorts of substrates. But I'll show you some data that we have that we published last year on asking whether or not epidermal cells sense the underlying bulk stiffness of their substrate. And this is work which was primarily carried out by Britta Chapman, who's now a postdoc in Boston at BU. So she took substrates made of PDMS, which is essentially a rubber material. She used a chemical cross-linker to immobilize extracellular matrix proteins. And then she made substrates which ranged in bulk stiffness from only 1 kilopascals up to 500 kilopascals. And the bulk stiffness of human dermis is roughly 5 kilopascals, so definitely at the soft end of this spectrum. So she plated the cells. These are again scanning EMs of single cells. And she noticed that actually the bulk stiffness of the substrate didn't affect whether or not the cells spread fully. That's shown quantitatively here, where she's measuring cell area along over here. And somewhat to our disappointment, we found that although there's a lot of literature saying that epithelial cells respond to bulk stiffness, we found that at 24 hours, the proportion of cells that differentiate was completely unaffected by the bulk stiffness of the substrate. So this was saying that an epidermal cell doesn't care if it's sitting on a soft substrate like connective tissue or if it's on glass or plastic. It doesn't read that as a factor that influences differentiation. We were a bit nervous about these results, though, because there's so many reports in the literature that cells do sense bulk stiffness. So we went back and we did the experiments again, but this time we did them using hydrogels. I'm just going to drop the lights, that's better. So now we're taking polyacrylamide hydrogels. We're again chemically coating the extracellular matrix, and we're again looking at a range of stiffnesses of the substrate. Now if we look by scanning EM at single cells, we see that on the softest substrates, the cells are attached but they're rounded, whereas on the stiff substrates, they're able to spread completely. The spreading is shown quantitatively over here. And now if we look at differentiation, we see that the cells which are on the soft substrates are much more likely to differentiate than on the stiff ones. So how does this work? If they're not reading bulk stiffness, are they responding to some other aspect of the surface? And Britta did a whole series of experiments, which I won't go through. But essentially, she found that when you change the bulk stiffness of polyacrylamide, you change the porosity of the surface. So this is illustrated in these two EMs here. So the surface of the hydrogels has a honeycomb structure, and the size of the pores in the honeycomb is much greater when the substrates are soft than when they're stiff. We also noticed that, of course, cells adhere to the extracellular matrix via extracellular matrix receptors called integrins, which are heterodimers. And we noticed that on the stiff hydrogels, they were able to cluster in focal adhesions, which you can see here. However, they couldn't do that on the soft substrates. So this led Willem Huck actually to propose that what the cells were reading when we plated them on the soft hydrogels was the fact that the collagen was anchored with spacing that was further apart than on the stiff substrates. So we envisaged the cells tugging on the extracellular matrix and measuring how much resistance there was to the tugging process. And so if that was the case, then we argued that if we kept the concentration of the polyacrylamide the same, but we changed the level of crosslinker, we should be able to control differentiation. And that was what we found. Here we're looking, keeping, as I said, the substrate stiffness constant. It's 20 kilopascals, which is a stiff substrate, but now having high or low concentrations of crosslinker. And sure enough, with a low crosslinker concentration, the cells are unable to spread, and within 24 hours they differentiate. Whereas if we have a high concentration of crosslinker, they spread and they don't differentiate. Now, we had expected that the way that this signal was received by the cell and translated into a process of differentiation would be the same as on the micropatterned islands. But in fact, that's not the case. And this is what I described previously for micropatterned islands. Here on the hydrogels, we think that what is happening is that signaling is going through this classic ERK-MAP kinase cascade. We know that AP1 transcription factor activity is important, but it's independent of a stimulation of SRF transcription. And so at the moment, Ajay Mishra in the lab is looking for a junk phosphatase, which we believe normally negatively regulates AP1 activity and is downstream of this pathway. So to summarize what I've said so far, we can do a lot of interesting experiments working with primary cultures of human epidermal cells. We can make environments in which we can precisely control different aspects of cell behavior. And we have shown that different external cues can trigger the cells to differentiate, but using different signaling pathways. And in the case of these soft substrates, we think that what is important is the mechanical force that the cells are exerting on the extracellular matrix. Now, before I leave the in vitro stripped down approach to the in vitro approach to looking at stem cells, I want to tell you briefly about another study we did, which was designed to look at intrinsic regulators of stem cells. And this is the work of Klaus Mulder, who now has his own group in Nijmegen. And what he wanted to do was to identify chromatin-modifying genes which might affect whether or not epidermal cells remain as stem cells. So he took the approach of knocking down 332 different chromatin regulators and performed a screen in which he looked at whether or not knocking down any of these factors promoted differentiation. We can do this very nicely, plating cells in 96 well plates or 384 well plates. What we do is we knock down the gene of interest, we stimulate differentiation, and then after 24 or 48 hours, we use our antibodies to differentiation markers to quantitate differentiation, and we can control for cell numbers. So this assay works very nicely. We've used it previously for drug screens as well. And then we can just relate the data on differentiation to the gene that we've knocked down. It sounds quite straightforward, but actually it's a massive project in terms of computational biology. And Klaas did a fantastic job of this in collaboration with Florian Markowitz's group. And the fruits of their labors are summarized in this slide here, where we've listed the different factors that we knocked down, the different differentiation stimuli that we used here, and then we've clustered the factors according to whether knockdown stimulates differentiation, which is in purple, or inhibits differentiation, which is down here in orange. And what Klaas did next was to focus on the genes which are required to keep the cells in the stem cell compartment and have a look bioinformatically for whether or not there are interactions between these different chromatin factors. Now, some of the factors that he found in this category are already known to regulate differentiation in the epidermis, and some of them are known to be in physical complexes with one another. But what was important from his studies was that he found factors which have the same function but are not physically associated with one another. And the other thing he found, which was really quite remarkable, was that even though these different factors regulate different genes, if you have a look at the functions of the genes, you find that the overwhelming majority of the genes that they regulate are control proliferation, so self-renewal of stem cells, or their ability to adhere to the underlying extracellular matrix. So by this screen for intrinsic regulators of differentiation, what Klaas came up with was an observation that the chromatin state of the cells influences the way the cells interact with the underlying extracellular matrix. And so we feel that by regulating different genes, but all of which have similar functions, you have an intrinsic way of protecting stem cells from undergoing differentiation. And we like the idea of this interplay between intrinsic and extrinsic signals. So those are all the experiments that I wanted to tell you about with our cells in culture. I've shown you what a beautifully simple system it is, how we can give very precise extrinsic signals or change the intrinsic state of the cell and get valuable information about differentiation. But of course, as I mentioned, there's much more to the skin than just the epidermis. And if you look at this histological section of the back skin of a mouse, you can see that I've just really been talking about this part here. And we have, in addition to the interfollicular epidermis, we have the sebaceous glands and the hair follicles. And so, as well as doing the work that we've done with human cells, we and many other groups have been interested in looking at the stem cell compartment in mammalian epidermis. And the sorts of approaches that are used for these studies are summarized on this slide here from Kai Kretschmar. One approach that you can take is to break the epidermis apart and interrogate the function of the cells by plating the cells in culture and looking at their ability to form actively grown clones. You can also take the skin apart but rebuild it on the back of a mouse. And a skin reconstitution assay that we use a lot is shown schematically here. We take an immune-compromised or syngeneic mouse, we make a full thickness wound in the back of the mouse, and then we implant into this wound a silicone rubber chamber which is shaped a bit like a hat. And then what we can do is inject different combinations of epidermal and dermal cells, and with the right combinations we can find within 5 weeks that we've reconstituted the skin completely with the hairs. These approaches are based on taking the tissue apart, but for some experiments you'd like to look at the situation in which the skin is undamaged. And there are lots of very useful genetic tools for looking at that. For example, there are ways in which you can monitor how frequently the cells are dividing in the epidermis. And it's well known now that there are different subpopulations of cells in the epidermis which divide rapidly or divide infrequently. And then the other approach, which I think is probably the most elegant of all, is to identify subsets of cells and then perform lineage tracing so that you can mark all of the daughter cells of the cell of interest, both in healthy skin and in disease, and build up some information about the properties of the tissue. Now, in an ideal world, all of these different approaches would have led to the identification of the one true stem cell in the epidermis. And I don't know whether it's because it's a contentious field or it's just life, but the way we are at the moment is that we have identified, and by we I mean many groups around the world, lots and lots of different subsets of epidermal stem cells, which express different markers, such as this one here, these ones all the way down here, and which are positioned in different regions of the epidermis. And the sense that I have of the situation for the different epidermal stem cell populations is one, really, it's a model that we proposed several years ago. But what we envisage is that there are multiple populations of stem cells. We know from skin reconstitution assays that if you take a stem cell out of context, it shows the ability to make all of the epidermal lineages. But we think that under steady state conditions, a stem cell lying here, for example, will only make the interfollicular epidermis. A stem cell in the lower part of the hair follicle will only make the hair follicle lineages, and that in the upper part of the hair follicle are the stem cells which primarily are making the sebaceous glands. But it is remarkable that these different subsets of epidermal stem cells show this ability to differentiate down all of the different lineages depending on the signals that they receive. Now, one pathway that we've been interested in for many years is the canonical Wnt signaling pathway. This is a pathway which operates when Wnt, which is an external signal, binds to its receptor complex, and as a result of that, the cytoplasmic protein beta-catenin is able to accumulate and translocate into the nucleus to regulate gene expression in combination with TCF-left transcription factors and other cofactors as well. And one reason that we have been very interested in this pathway is that it has a remarkable effect when you activate it in adult epidermis. So here I'm just going to introduce you to a technique that we developed 10 years ago now, which allows us to look at large areas of the epidermis. And this is a whole mount labeling technique, where essentially we take the tail skin of a mouse and we separate the epidermis from the dermis as an intact sheet. And then we fix and label it with antibodies, and then we can image the epidermis on a confocal microscope by laying this sheet onto the slide. So you're looking at the epidermis from the perspective of the dermis or the basement membrane. And in the tail skin of the mouse, the hair follicles are arranged in groups of 3. They're these structures like icicles that you can see here. They have prominent sebaceous glands, which are these sac-like structures, and the hair follicles are embedded in the interfollicular epidermis. So we can use this technique to rapidly monitor how the epidermis responds when you activate the Wnt pathway. And the mouse that we use, a transgenic mouse in which to activate this pathway, is shown schematically here. We target all the stem cells using the keratin 14 promoter. We express a stabilized form of beta-catenin, but in order to selectively activate beta-catenin in the adult, the C-terminus of beta-catenin is fused to a mutant estrogen receptor. So if you take the drug 4-hydroxytamoxifen and paint it on the surface of the skin, you can control where and when and for how long the pathway is activated. Now, hair follicles go through cycles of growth and rest. And in this example here, the hair follicles look quite short because they're in the resting phase of the hair cycle. If you apply tamoxifen to activate the Wnt pathway, you stimulate existing follicles to elongate and grow, so they become these massive structures which you can see here. But in addition, instead of making sebaceous glands or interfollicular epidermis, you reprogram the rest of the epidermis, so now it's committed to making new hair follicles. And all of these little red lumps that you can see represent attempts by the skin to make new hair follicles. So these changes in the epidermis are so remarkable and so radical that it really does beg the question of what is happening in the underlying connective tissue. Is there any evidence that you can change the epidermis without the connective tissue being remodeled? Or in fact, do these changes in the epidermis cause a plasticity within the underlying connective tissue? So you've seen this slide before, but now I just want to highlight the different regions of the connective tissue that we're interested in. So the connective tissue is the dermis. It has an upper region called the papillary dermis and a deeper dermis called the reticular dermis. The difference between these two regions is primarily that the density of fibroblasts is higher here. There's a lower density of blood vessels. And down here is where you get the bulk of the fibrillar collagen and elastin. Underneath this is a region called the hypodermis, which contains mature differentiated fat cells, and it also has precursors of fat cells. And then other structures which are very interesting in the dermis are this smooth muscle here called the erector pili muscle, which causes hair follicles to stand on end, for example, when the animal is cold. And down here at the base of the hair follicle are specialized fibroblasts called dermal papilla cells, which we believe are captured by the overlying epidermis and then communicate exclusively with the neighboring epithelial cells to regulate the hair growth cycle. So what happens to the dermis when you remodel the epidermis by activating the Wnt pathway? Well, a tool that has proved very valuable for looking at this is to look at mice in which the PDGF receptor alpha promoter drives expression of nuclear GFP. So in this example here, it's a cross-section of skin in a newborn mouse. The epithelium is labeled in red with keratin 14. But if you look for where the GFP positive cells are, you can beautifully pick out the nuclei of all of the fibroblasts and the fat cells in all of the regions of the skin. And so if we cross these mice with the beta-catenin mouse, we can have a look at how the dermis responds to changes in epidermal gene expression. This is a heat map where red is upregulated genes and blue is downregulated genes. And we're just looking here at triplicate mice, where these are the fibroblasts from these triplicate mice. And we're looking at adult mice where the hair follicle is in the resting stage of the cycle. Or if we apply one dose of tamoxifen to drive cells into anagen, or if we activate the beta-catenin pathway more extensively and induce ectopic follicles. And then we've compared these three types of adult fibroblasts with fibroblasts that we've isolated from newborn skin. And what was really striking to us, just looking at this pattern, is that when we remodel the epidermis, the overall appearance of the dermis comes to resemble that of neonatal dermis more closely than the other adult fibroblasts. So this got us wondering whether, in fact, activating beta-catenin in the epidermis was rejuvenating the dermis, making it more like newborn skin. There are a lot of different Go terms which came up in the arrays, but what was really striking was that we got an upregulation of genes associated with proliferation and a downregulation of genes associated with the extracellular matrix. So we were able to go from this gene ontology analysis back to the skin, and now instead of looking at the epidermis, we looked at the dermis and asked what was going on. And sure enough, we saw exactly what we would have predicted from the gene expression profiles. So if you just look at the green nuclei up here, you can see that when we reprogram the epidermis and make new hair follicles, there's a big increase in the density of the fibroblasts. Now the density comes to resemble newborn skin, and it has much more fibroblasts than in the adult situation. And then we used the classic histochemical stain, which stains mature collagen purple. And now you can see that comparing adult skin with neonatal skin, in neonatal skin, the extracellular matrix is less mature. And we see that when we reprogram the epidermis, we also change and cause remodeling of the extracellular matrix in the dermis. So having seen these changes, I'm going to now just take you through some of our unpublished data in which we have looked specifically, first of all, at the fat cell layer in the skin, and then more globally at different populations of fibroblasts. So it has been known for a very long time that if you look at the back skin of a mouse from the day it is born up through until it's several months old, the hair follicles go through cycles of growth and rest and growth and rest. And associated with that, the fat cell layer of the skin also increases in size as the follicles grows down, then it reduces in size, it increases in size, and so on. Now, to our embarrassment, we had in the lab for 10 years some transgenic mice in which we had targeted the Wnt pathway in the epidermis, and we had a very striking fat phenotype. We had sort of noticed that mice tend to get fatter as they get old, and that our mice were a bit skinny, but then since our mice also were lacking hair follicles, we thought that probably they just looked skinny because they didn't have hairs. But this is a mouse in which we inhibit Wnt signaling with a dominant negative LEF1 transcription factor. And if you look at the size of the fat layer in an old mouse in the wild type versus this mouse where we've blocked epidermal Wnt signaling, you can see that there's far fewer fat cells. Conversely, if we activate the Wnt pathway, which we can do either in adult mouse with our beta-catenin ER model or in the embryo, which is shown down here, in both cases, activating epidermal Wnt signaling gives us a big expansion of the fat layer. This antibody marker shows mature fat cells. You can just see the green has increased here relative to here. And then this is an example of wild type and transgenic mice where the red labeling shows you mature fat. And you can see this big increase in the fat layer when we activate epidermal Wnt signaling. So how is this going on? I've told you that we're interested in the signals that regulate the epidermis, but now it would seem that changing the epidermis is providing signals that regulate the dermis. And Giacomo Donati and Hiro Fujiwara, who did these experiments, took a cell line called 3T3L1 cells, which are known to be able, they're derived from embryonic fibroblasts, and they're known to be able to differentiate into fat cells. So what Giacomo did was to say, if I take conditioned medium from cultured epidermal cells, can I influence whether or not the cells differentiate into fat? And the answer is yes, you can. So this brown staining that you can see here represents fat differentiation. If the conditioned medium comes from cells in which the Wnt pathway is activated, you get more fat. And if it comes from keratinocytes in which the pathway is inhibited, you see less fat. Very recently, Hiro and Giacomo have made some progress in identifying some of the factors that are important. And again, if we just look at the labeling that you can see here, we see, for example, BMPs, in particular BMP6 and BMP2, can strongly stimulate adipocyte differentiation. And then we found that IGF-2, which is part of the insulin signaling cascade, stimulates differentiation. And if you look here, we're looking at stimulation of fat differentiation by different concentrations of insulin. And these are the wild-type keratinocytes. These were those in which the beta-catenin pathway is activated. And you see that the wild-type cells are much more sensitive to insulin than the beta-catenin cells, which are very good at stimulating differentiation. So the model that we're working on at the moment is one in which reciprocal signaling between the hair follicles and the fat layer are important in coordinating the hair growth cycle with the thickness of the adipocyte layer. So now I want to end with another recent story. And this is work which has been led by Ryan Driscoll in the lab, but with a lot of significant contributions from other members of the lab. And that is to say, well, what do we know about fibroblasts in the connective tissue? Is it that they are all the same? People will often say, oh, I just used some fibroblasts to do this, or this is how fibroblasts behave. But if you look at the structure of the skin, as I've shown you, the connective tissue, it looks like the fibroblasts have different properties in different regions. So we asked whether we could identify different subpopulations of fibroblasts. And the two challenges that we faced is that when you do lineage tracing of fibroblasts, they don't form readily identifiable clones that you could work with. And also there are really very few markers which could be used to distinguish different potential subsets of fibroblasts. So the first approach that Ryan took was to look at the dermis at different stages of embryonic development. So at E12.5, the epidermis is about 1 or 2 layers thick, and the dermis looks relatively unstructured. But if you go out to P2, so that's 2 days after birth, all of the epidermal structures are developing, and now you can see a fat layer, you can see the dermal papilla, you can see papillary and reticular dermis. And then he took antibodies to a range of candidate markers of fibroblast subpopulations, labeled at all of these different stages of development, and then asked whether any of the markers change as a function of time or layer. And this is a sort of digital tissue microarray, where what he's done is to take boxes from the confocal images that are representative of upper or lower dermis, and then arrayed them as a function of time. So PDGF receptor alpha is always expressed by fibroblasts at all stages of development. E-cadherin is a marker of epidermal cells, so it's never expressed. And then as you look down through these different markers, you can see some which show quite dynamic patterns of expression. And so what Ryan did next was to take some of these markers, which are to cell surface antigens, and use those in the combination with PDGF receptor alpha GFP to sort out different subsets of fibroblasts. So this shows you the experiments that he did. This is the skin reconstitution assay that I mentioned. We're taking an excess of unlabeled epidermis and unfractionated dermis, and then we're spiking in GFP positive fibroblast subsets. We let the skin form, and then we look at where we can pick up green fibroblasts. So do we find them throughout the dermis, or do we see them restricted to different regions? So there are markers that we have which enrich for upperdermal fibroblasts, such as expression of CD26 and lack of SCAR1. And in that situation, we can easily pick them up in the upper dermis, but we tend not to see them in the lower dermis. So that's this example of this blue bar here and here. Conversely, we have markers which, where the cells contribute to lower dermis, which is shown here. These are SCAR1 positive and lack a marker called DLK1, but we don't see them in the upper dermis, so that's the red bars here. We do have evidence for fibroblasts which can contribute to all regions of the dermis, but actually the markers that they express, which are DLK1 and SCAR1, the DLK1 marker doesn't persist in adult skin. So there could be multipotent fibroblasts resident in the dermis, but we can't find them with the markers we're using at the moment. So it looks from these skin reconstitution assays as though there are different subsets of fibroblasts with different properties. But is that functionally important? Well, we did a similar experimental construct here, but in this case, what Ryan did was to use flow cytometry to sort out the populations of cells that were of interest. And the main finding here was that if we depleted cells which have characteristics of upper dermis, we now failed to make hair follicles in the skin reconstitution assays. Whereas if we took out the lower dermal fibroblasts, there was no effect at all. Now, I told you that there are pros and cons of these different sorts of assays. And of course, the concern is that in skin reconstitution, you're seeing what cells can do, but not what they do normally. And so for the last few experiments that I'm going to describe, what Ryan has done is to use lineage tracing approaches. So he's taken the markers that he identified from the tissue arrays and used those for lineage tracing in undamaged skin. So one of the markers that is interesting is DLK1. And that's interesting because at E12.5 of development, it's expressed by all fibroblasts, but at E16.5, it's only expressed by lower dermal cells. So what we can do is take a DLK1 mouse expressing CRE-ER, cross it with a mouse which will express TD tomato when the cell is exposed to CRE to remove the stop codons. And then we can look at different stages after birth and see where we can find cells which are labeled red. So if we label at E12.5, you can see that we can pick up red labeled cells in the all of the different regions of the dermis. In particular, we see them in the erector pili muscles, we see them in the dermal papillae, and we see them down here in the fat layer. In contrast, if we label it E16.5, we're only seeing a contribution to the lower dermis and not to the upper dermis. So it would seem that at between E12.5 and E16.5, something happens, a fate switch, so that these cells become committed to the lower dermal lineages. We have a number of different markers of the upper dermis. In particular, we can use BLIMP1 CRE or we can use LRIG1 CRE-ER, and we get results which are, again, consistent with a differentiation decision being made. So if BLIMP1 is only expressed transiently in the upper dermis, and we see beautiful evidence for selective labeling of the upper dermis, and in particular evidence that the erector pili muscles are solely derived from the papillary dermis. And here with LRIG1 CRE-ER, you can see again that we're labeling the upper dermis and not the lower dermis. So we believe that what is happening is as the dermis develops, at this stage of development, something happens which means that these upper dermal fibroblasts are different from these lower dermal fibroblasts. And so what we've gone on to do is to ask whether we can modulate the abundance of these fibroblasts, and if so, whether it impacts on wound healing. So we know that fibroblasts are activated when the skin is wounded. And we first of all asked, do the upper and lower dermal fibroblasts contribute equally to wound repair? So this is a full thickness wound, and we can analyze our labeled populations at different times after making the wound. So what we found from these studies is that the initial wave of wound repair is solely contributed by lower dermal fibroblasts that we can label with DLK1 CRE-ER. We see a big influx of these cells into the open wound, and these are the cells which express markers of activated fibroblasts, myofibroblasts. If we look what happens with the upper dermal lineage, these cells, the fibroblasts, only repopulate the wound much later. In fact, largely after the wound has re-epithelialized, and we can start to pick the cells coming up slowly underneath the epidermis. If we activate the Wnt pathway in the epidermis, again using our favorite K14 beta-catenin ER model, we find that we expand both the upper and the lower dermal lineages. The quantitation by flow cytometry is shown here. So this is where we've made ectopic hair follicles. We see a significant increase in upper dermal fibroblasts and lower dermal fibroblasts. But what happens to the function of skin repair if we cross our mice with the beta-catenin model? And this is work which was done by Beate Lichtenberger. So what Beate found was that if the mice were treated with tamoxifen to activate the Wnt pathway and then subsequently wounded, so we've expanded the upper dermal lineage, now we see, and this you can pick this up as these green spots down here, we see a much more robust contribution of the upper dermal fibroblasts to the wound. And to our delight, now instead of the wounds being devoid of hair follicles, we can pick up new hair follicles forming in the wounds. So this is the quantitation of ectopic hair follicle formation. This is the control. This is where we've activated beta-catenin with 1 dose of tamoxifen, or here with repeated doses so we can see the hair follicles coming up. So to summarize these last experiments, by turning our focus away from the epidermis and looking now in the dermis, we see evidence that there are lineage relationships within the dermis, such that there are at least 2 distinct lineages. And we think that the lower dermal cells are first to repopulate the wounds, but it's the upper dermal cells which are required for hair follicle formation. And of course, there are many questions now that we would like to be able to address about the properties and potential of these different subsets of fibroblasts in the skin. So with that, I'd like to thank you for listening, and I'd like to especially say thank you to a large number of people who've contributed to the work over the years. So John, Britta and Klaas did the work that I described with culturing human keratinocytes. Charlotte Collins really started off our interest in how activating the Wnt pathway in the epidermis would reprogram the dermis. Hiro Fujiwara I mentioned because he now has his own group in Kobe in Japan, but Beata, Esther, Kai, Giacomo and Ryan are all in the lab presently, and they've done a fantastic job in characterizing the different fibroblast lineages. So with that, I'm happy to take questions.